Michele Petrillo turn with dendrochronology of the Azorian juniper and Juniperus brevifolia.
Thank you. Are you right? One, two, three, test. Yeah, it works. Okay, so for today's presentation, I decided to talk about the thesis that I carried out for my bachelor. This was 2 years ago. It was on a dendrochronological study on the Juniperus brevifolia, or commonly known as Azorian juniper. Just to clarify, the title is not a critique to the university system, but you'll understand in a few slides. So, yeah, a bit of general background. Back then, I was studying in the Netherlands in a university called Avans University. I was studying environmental sciences, and for my thesis, I decided to go to the Azores and work with a research team called Modelandis, with the supervision of Professor Luis Silva. For who doesn't know, the Azores is an archipelago of islands in the middle of the Atlantic. It's composed of 9 islands, and I was mainly working in the island of San Miguel. The study was on Juniperus brevifolia, which is a plant that is endemic to the Azores. These islands, as I said, are in the middle of the Atlantic. They're isolated, and so they have a high proportion of endemism. According to the red list of the IUCN, it is considered a vulnerable species. So, yeah, this is a presentation that I actually already have done for my thesis. I mean, the formal goal, I already presented, the results of the formal goals I already presented for my bachelor thesis. So today, I would like to present something different. Aside of the research that I didn't get the chance to expose back then. Well, you can see like on the board that the original goal was to preserve and valorize the Azorian juniper by building a master chronology, evaluating its potential for climate reconstruction, creating models to estimate its age based on dendromatic traits such as height or the diameter at the base, and studying the wood anatomy. But when I was working on this study, I also have done personal observations that I didn't get the chance to expose. These were mostly focused on evolutionary adaptations of the plant, on the biology in general of the plant that could give insights in the field of biomimicry. For who doesn't know, biomimicry is a discipline that looks at nature, at patterns that you find in nature to find insights for design in the nowadays world for products, construction, architecture, etc. So starting from the macroscopic observations during the fieldwork, I made some observation that I will now explain. Starting from some pictures that I took of the plant, you can see on the top left corner the berries. You can see the leaves that are very short and needle-like of the plant on the left bottom corner. Yeah, you can see also the trunk that is very irregular, and I mean, at least for the people that are from Europe, you probably know the Juniperus communis, which is very much related to this one of the Azores. It's just that because the plant doesn't have a competition on the island, because there's only like a limited amount of different species of trees. On these islands, the Azorian juniper manages to grow as a tree and not as a shrub, reaching also heights of 8 meters, which for me was kind of strange when I first got there. I was used to the shrubs of the juniper in Europe. Before moving on, no, actually, yeah. Let's go directly to the first observation. The first observation that I made was on the reaction wood, which is called compression wood for the Juniperus brevifolia, since it's a gymnosperm. What I noticed in the wood cores was that when the juniper was growing on a slope terrain, to adjust the tilting and to keep the vertical alignment, it was growing this compression wood on the downside, as you can see also in the diagram. Well, this is opposite to the angiosperms, which grow tension wood on the upper side. I do not know why the technique used for angiosperms and gymnosperms differs. Like they're practically opposite, and so this could be itself an interesting research question. But the idea that I had was to emulate this cell structure that is different in the compression wood compared to the opposite wood on the A side for architecture and product design. So whenever you have artificial or natural tilting in buildings, perhaps the structure used, the technique, the strategy used by the gymnosperms could be used to provide stability, and the same for products that need resistance despite having tilting in their shape. Then the second macroscopic observation that I made was on the leaf canopy. Okay, 3 minutes left. The leaves are needle-like and short. They are probably a strategy to prevent grazing, and they have a downward configuration, which I think doesn't influence, which has a low influence on the trajectory of the raindrops. So compared to other plants that use, for instance, the broad leaves to distribute the water further away from the trunk for roots outward development, this plant doesn't do it, and this is just an observation. I didn't go further to make a possible insight for design. Then for the microscopic observations, I did some sampling using a trefoil, making microcores, which I then analyzed under the light microscope. Well, the first observation was on the growth rings. There is a clear difference between the early wood and late wood cells. The late wood cells, which grow after the growing season, they are smaller and they have a thicker cell wall, which provides, they provide a shield for the precious wood. And I believe that this strategy, this pattern, this design could be used for insulating material in construction or packaging. Then here are some pictures of the wood anatomy taken under the light microscope, which I will skip because I don't have a lot of time. I will go straight to the observation that I made on the parenchyma and the axial, parenchyma, radial, and axial cells. Everybody knows, it's commonly known that there is a transport from bottom up and from up down in the trees through the trachate cells. What is less studied is the axial and radial transport of nutrients through the parenchyma cells. From what I observed under the microscope, I sort of got the idea that there is a pattern, as you see in the diagram here, where you have the parenchyma cells providing an alternative transport of nutrients. Then there was another slide on the auxin model, but I don't have enough time because the time is over. So I will skip this one, and I will just ask you if you have any feedback, but I guess that this will be for later during the questions. The idea was to share the ideas and observations that I made, so to, talk about it together and brainstorm together and have like a fun exercise to do. Yeah, that was it.